by looking at this paper, novel histone deacetylase inhibitor CG200745 induces clonogenic cell death by modulating the acetylation of p53 in cancer cells. That is an exciting title. I don't know what it is. So we looked, if you recall, back in the p53 chapter, we did see aspects of acetylation and deacetylases involved in the whole p53 story. So they're looking at using that part of the story sort of as a possible therapeutic intervention in certain types of cancer. And so they designed some kind of inhibitor of a histone deacetylase. So we're going to be looking at some histone stuff, some p53 stuff, and some p21 stuff in here. So, I trust you right through this. So some highlights from the introduction. Okay, so we've already seen in our own studies that histone deacetylases are involved in that part of the p53 story, so therefore have some link to the onset and progression of cancer. So HDAC, so histone deacetylases are HDAC inhibitors. Then if we inhibit the deacetylation, so D is off, right? So we're taking something off. So if we inhibit the things taken off the acetyl groups, that means the acetyl groups stay there. So whatever the targets of these things are remain hyperacetylated. Okay, and other people have shown that among the hyperacetylated things are p53, among other proteins, as we'll see. And so therefore, HDAC inhibitors have been suggested then as anti-cancer therapeutics. And at least at the time of this paper, which is in 2010, there were a number of them in phase 1 and phase 2 clinical trials. Phase 1 clinical trials usually a small safety trial, so you make sure that the thing isn't killing people, literally. Phase 2 are sort of the larger trials that you're always looking at safety, but the phase 2 trials are where you're really looking at efficacy. So it does the drug that you've shown to be relatively safe, does it do anything helpful. Okay, so in general, HDAC inhibitors have been shown to invoke cell cycle arrest, induction of apoptosis, induction of differentiation, all very good anti-proliferative kinds of things, right? So if we're arresting the cell cycle, then we're slowing things down. Apoptosis, we're actively killing cells. If you reduce differentiation, right, if differentiation goes up, what tends to go down?
Proliferation.
Is inversely proportional to differentiation. Proliferation. Okay. So they're suggesting that this all might be working through, so if you're not pulling them off, then we're leaving certain things hyperacetylated, like histone proteins and other non-histone proteins like p53, transcription factors like p53. And therefore, both of those changes are going to then change gene expression patterns, hopefully in an anti-proliferative type way. So that's kind of what they're going to look at in this study. And so somebody came up with this particular drug, and we'll see a picture of it in a second. And they're going to look at the effects of this particular HDAC inhibitor on p53 specifically, among other things, but that's going to be one of the main, obviously the main targets of this study. And they're suggesting that hyperacetylated p53 is stabilized, okay, in that it's not going to get ubiquitinated, therefore not destroyed. Remember the whole MDM2 kind of story with p53. And therefore, if p53 is still around and it's still active and not being actively destroyed, then we have that circuit to potentially get rid of the cancer cell through apoptosis. So it's a nice synthetic kind of story here, so I thought it fit well with this section of the course. Okay, so the first figure is just showing us what the drug looks like, so if you're interested, that's what GC 2007-45 looks like. I'm just going to call it GC, I think, for now, or CG. Kind of looks like. Okay, so they're working here in this first experiment with cancer or colorectal cancer cells called RKO cells. So this first experiment here is sort of a time course kind of experiment. So they're exposing these cells to this drug for these varying amounts of hours, so units are hours here. Take the cells, extract the protein, run them on the gel. They have and they have an antibody against in this experiment here, histone H3, specifically acetylated histone H3. You can make antibodies against even derivatized types of proteins as well. And the beta-actin is just a control that shouldn't be affected by any HDAC inhibitor. All right, so that is pretty constant. So it's both a control for the drug and a loading control, basically, for the experiment, so that equal amounts of protein have been loaded into each of these lanes. So if you see more in the experimental lane, you know that that's real. So, having said all that, what can we conclude then over this time course as we expose the cells to this drug? What happens to the amount of acetylated histone H3?
Increases.
It's pretty clear, right? So, without the drug at time 0, there's nothing. By the time we get to 24 hours exposure, this is a constant exposure. Do we know the concentration? Yes. So this was 10 micromolar concentration of the drug. Okay, so it's a constant concentration, but over time we see an increase in the amount of acetylated histone H3. And if you know anything about this, if you add acetyl groups to histones, that tends to change the properties of the nucleosome and the ability of DNA to wrap around it and that kind of thing, so it does have an effect on gene expression. Okay, very good. Okay, so it seems that over time, in the presence of this drug, the acetyl groups sort of build up on histones, or this particular histone at least. And where's H3, by the way? Part of the nucleosome or part of the linker?
Nucleosome.
It's part of the nucleosome, right?
One of the subunits.
2A, 2B, 3, and 4 make up the core octamer. Right, so this is part of the nucleosome that the DNA is wrapped around. Okay, good. And I probably would have presented this one first. I'm going to put this one as B, I think, and this one as C. But in any event, the next experiment they do kind of the opposite, same amount of time, but different concentrations of the drug. Like I said, I would have shown that one first. But in any event. So here it's not time. Everything's been treated for 16 hours. But we have exposed the cells to different concentrations. So this is a micromolar. So 0, 0.11, and 10 micromolar Cg. And then this other drug, SAHA, is another known histone deacetylase inhibitor. Okay, so they're basically comparing the new drug to this known drug. Again, we're looking at the amount of acetylated histone H3. Okay, the control is very nice. That looks all nice and even. So what do we see here?
Looks like an increase, but the SAHA
Less
is
effective.
showing off. It looks like it decreases there too. Unless I'm just.
Well, like that's a constant. There's only one amount of that drug.
Oh, okay.
Right,
Then in that case, it looks like it's increasing.
I didn't write down what the concentration of the other drug was, but yeah, I think there's a clear increase versus CG concentration. So at 0 and 0.1, there's really not very much acetylated histone. Why not? What's the drug do?
Because it inhibits the deacetylase.
Yes, the deacetylase. So if we're not inhibiting the deacetylase, what do deacetylases do? Pull acetyl groups off of histones. Okay, so if we're not inhibiting it very much, then the deacetylases work. We're pulling the acetyl groups off of the histones, and therefore there's not much acetylated histone now. But as we start inhibiting the deacetylases with increasing concentrations of drug, more of the acetyl histone starts showing up. I think that kind of makes sense. And it looks like, just kind of sort of eyeballing it, that looks sort of in between the 1 micromolar and the 10 micromolar dose of the new drug. I kind of eyeball of that. They don't quantitate it, so I can't really say, but sort of just eyeballing it, looks like there's more than the 1, but less than the 10. Okay, so it looks like this thing is working the way it's supposed to. It certainly looks like it's working as a deacetylase. And so as you expose either to higher concentrations, so then I think for the rest of the experiments, I think they use the 10 micromolar dose. That's why I'm going to show that really next. And then using that 10 micromolar dose, they show this as a time course. Okay, so over time, at this dose, it seems to deacetylate these histones. Make sense? Okay, so in the second experiment, they're looking at, instead of the histone, they're starting to look at expression levels of various other players. So we see p53, MDM2, p21, CYP1. Okay, so we already know what p53 is, right? The central thing. MDM2 is that protein that interacts with it and usually ends up causing p53's destruction. And then remember, what's CYP1?
Cyclin.
Way back from
They did with cyclins.
Right, so the cyclin inhibitor, pre or post R point.
Post.
Right, this is one of the post R point inhibitors. Okay, good. And again, are active control, so this all looks, at least eyeballing it, looks, not all looks even. So again, they're doing this as a time course, so this is in hours at the 10 micromolar, and like I said, I think that's what they're going to use for the rest of the time. So all of this is the CG drug at 10 micromolar. Here's the one lane of some amount of the other sort of comparator HDAC. So let's just kind of work our way through. So what's happening to P53 over time of exposure to this drug?
Concentration increases. It's going to increase.
Over the entire time course, or?
No, right around 16, it looks like it falls off.
Yeah, kind of looks like it's peaking around 8 and then starts to go back down. Okay, that's interesting. How about MDM2?
Looks the same as the increase.
Yeah, I think that's kind of following the same story. It's going to be increasing, quite peaks at exactly the same time, but kind of still peaks at 8 and then starts going down again. And it seems to be sort of the same thing here. And then how about P21 now, or CYP1? It's called CYP1.
The increases but much later.
Yeah, it's kind of the opposite. There's kind of nothing. While these two things are increasing, there's kind of nothing. And then while these are decreasing to some extent, that's when they kind of show up. It's kind of
What have to do with the R point?
I don't know what else to do. So, but we'll just note the observation for now. See if we can piece this together.
Based on the P53 increase, is this ideal at 8 or at around that time, or?
I don't understand your question.
Well, it increases, it peaks at 8, right, then kind of
Seems to.
decreases.
Kind of decreases, right? That's what we just said, right?
Why does that happen?
That's what we're going to work through, aren't we?
We'll come down.
I hope you already found out because I hope you all read this paper. So for now, we just have the observation. Okay. I guess one other thing we should note, what's sort of the relationship among these three proteins in that is, I asked the question without giving you the answer. Well, going back, what does P53 do? Well, most fundamentally, what is it? Well, yes and yes, but what most fundamentally is the P53 protein? What is its most direct job?
Filling the cell in response to stressors.
Okay, how does it do all of the things that you just told me? What does it most fundamentally do? These signals. The same test isn't Monday. Read me bullet number 2. It's a core short-term memory. Read me out loud.
So
Bullet number 2.
Factor responsible for apoptosis.
Pardon me?
In essence, what you're trying to get us to say is that P53 is a transcription factor responsible for apoptosis.
Actually, I really just wanted the first part of that. It is a transcription factor. Right? And it's its most fundamental identity. As a result of it being a transcription factor, it does all of these wonderful things that all of you mentioned. But it does that in light of its innate existential existence as a transcription factor. Right? So, with that in mind, let's go back here. What is the relationship between the transcription factor P53 and MDM2, and the genes for MDM2 and SIP1, as we looked at back in whatever the P53 chapter was? What chapter was that?
No.
Was it 9?
Yeah, not.
When showing MDM2, the answer will be the same for MDM2 and SIP1.
Why is the P53
To be created.
for apoptosis?
It's true, but I want to know the relationship between the transcription factor, P53, and the gene that encodes MDM2.
Positive feedback in the gene?
What is that telling us?
P53 is the transcription factor to activate MDM2, which then acts as a positive feedback.
There we
to
go.
turn it off.
Right, transcription factor. What do transcription factors do? They bind to genes and they turn them on, usually on, but sometimes off. This one clearly is turning it on, right? And we saw that that was sort of the crazy relationship between the two, right? P53 controls the gene that makes the protein that destroys P53, or that indirectly destroys P53, right? Connections, right? That's what 400 level courses are all about, making connections. Right? So we need to connect basic stuff we learned in the chapter to something in real life, if we want to use that terrible cliche. Okay, so papers real life, here's the basis. Right? So P53 hops onto the gene for MDM2 and typically turns it on. So, and since I don't have a picture of it, I thought there was in here, but apparently not. The same thing is true for SIP1. Thought there was a picture, but I do remember talking about that in that chapter. So, let's connect that to this. So as we make more P53,
Like we're turning on MDM2.
what would make sense?
We're turning on MDM2 and
Right.
P20.
And if we kind of look, it's, you know, a little bit behind in a sense. Right? So as we make more P53, we start making more MDM2 kind of really fast, and then for some other temporal reason, it doesn't turn on the CYP1 gene until much later, but it seems to turn that on as well. So there's an interesting connection among these three proteins, and that's probably why, in part, they chose them for this experiment. Making sense? We're making the connections now. Yes, we are, or? Okay. 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 So, the next two are sort of an attempt to quantify, in a way, this same phenomenon. So we're going to kind of see the same general kind of result here, but just in a different way. So what they did here was to make a plasmid, it's a little piece of DNA, that has a gene for an enzyme called luciferase, which when active, so when the gene is active, it makes luciferase protein, and that enzyme creates light. Okay, so you can put in this, it's essentially a spectrophotometer or something akin to that, and measure how much light is produced. So they put the gene under the control of either, in experiment B, a p53 response element. Okay, so if there's p53 around, it hops onto the p53 response element, which turns on the gene. Okay, so this will get transcribed and translated. We have the enzyme. With the enzyme, you have light. Okay, and that's what they're measuring here. It's a fold difference. Okay, so no drug, 10 micromolar drug. And in part C, same idea, except the response element here is for P21, or for CYP1. What do you call it? CYP1. So same idea, I'll just put it on the other side. So they expose the cells to the drug, and now and the cells have had this plasmid given to the cells. Okay, so they're a little genetically altered there, but that's okay. And then ask what effect does it have with those throwing the drug in? Well, we can kind of correlate this with that. So we've already seen if we throw the drug in, and this must have been a constant amount of time. 24 hours. Okay, so 24 hours, we have increased the amount of p53. It's on the decline here, but there's still p53 around. And now there's lots of CYP1 around. So it kind of makes sense if you understand that. This is just another way of showing kind of the same thing. So if there's lots of p53 around, then what's going to happen given that construct?
We're going to express more, it's going to light up.
Yeah, so the more p53 is going to pop onto here and turn on the gene, and that's what we see. And how about p21? Same idea, right? In 24 hours, we have a lot of p21 around, pops onto the red one, turns on the gene, makes a lot of light, we measure the light, and it's, in both cases, almost twofold higher amounts of light with the drug than without the drug. So it kind of correlates with what we see up here in terms of amounts of protein being made. This just kind of confirms that. This is as good an experiment, but it is what it is. That make sense? So that's all they're showing there. Okay. Okay, so far we've seen now that in the presence of the drug, histones keep their acetyl groups. We see an increase in the amounts of these three proteins. And let's see what we're doing here. And here, in this experiment, they're looking at now the death effect, in a sense, of presumably the changed expression of these proteins. So we have two types of cells here, still the RKO cells, so they're the original colon cancer cells. And then they have a sort of subline from that called RC 10.1. And the difference is RKO cells have P53, have normal P53, and the RC10 subline does not. So let's put that up here so you remember. So RKO is P53 positive, and the RC10.1 is P53 negative. So one makes it, one doesn't. Okay, and they're doing this in a dose-dependent manner, so here's the dose of the drug on the x-axis, and how many cells survive, okay, on the y-axis. So part A is the new drug, is the CG 2007-45 drug. Okay, and part B, I think, is the other one. Yeah, it's the comparator, is this SAH drug. Okay, so what do we see here with the new drug, with the CG drug?
More dosage, they both decrease.
Hmm?
And it seems like that when you have P53, that decreases. More susceptible. It's more susceptible.
I would agree totally with that. Okay, so both lines are going down, right? But the open symbols, the RKO ones, the P53 positive cells, seem to be dying, more of them seem to be dying, it's probably a simple way to say it, than the negative cells, the P53 negative cells. Which, again, if we start making connections, can make some sense. Right?
That
'Cause-
signals that P53
Right.
signals that, so.
right, as you've told me several times already this afternoon, right, P53 leads to cell death. So we have it around, and we're making more of it, apparently in the presence of this drug, then we should see more death, which is exactly what we're seeing. But we're still seeing death even without it, so that's kind of an interesting observation too. But we also know that there are other ways to kill cells, okay, other than P53 sponsored apoptosis. Okay, and the B part is the other drug, that SAHA drug. So we see, what do we see there?
It's the opposite. It's the opposite.
Yeah, it's kind of the opposite, and I don't think it's quite as good. Well, it looks almost as good with the negative ones. They're about this, they die to about the same extent, but the positive ones don't die to the same extent. Yeah, but it seems the opposite. So the SAHA HDAC, that's right, seems to have the opposite effect on these two cell types. I don't know what that means, but there's the observation. That makes sense, or we can see that's what those figures are saying. Okay. So now part of the question then is, okay, so we have this observation now where the drug seems to somehow increase the amount of P53, and now we're thinking maybe that increased P53 is inducing apoptosis more, which is exactly what it's supposed to do. So how is it kind of doing that? So this experiment is looking at how long the P53 sort of lasts. Okay, so remember, it has, under normal circumstances, a pretty rapid half-life, right? It should only last like 20 minutes or so. And so that's what they're kind of looking at here. So we have time across here, so we do this over an hour. So that's in minutes. They throw in, so this is an 8-hour exposure to the drug. CXH stands for cyclohexamide. We've talked about that drug before. Oh, that's, okay. You remember what cyclohexamide does? We've talked about it here. Say
Is
again?
this all in the G0, please?
I don't think so. I don't know. It's a translation inhibitor. It stops protein synthesis. So basically, in the presence of cyclohexamide, you can't make any new protein, is the simple idea. So whatever protein you're seeing here is not going to be new protein, so you can watch it. Whatever protein is there kind of disappear, is kind of the idea. So left side is just drug and cyclohexamide, right side is drug, cyclohexamide, and, no, I'm sorry. No, wait. Something doesn't make sense. Oh, I'm sorry. So both sides were treated with the drug for 8 hours to maximize p53 expression. Then they were treated with cyclohexamide, so no new drug, or no new p53. Then they took the drug away from here and they kept the drug there. That's how it's set up, sorry about that. So, and again, the active metal looks pretty even. So whatever we see here will seem to be real. So what do we see?
They look pretty similar. They look pretty similar.
Pretty similar, but exactly similar.
Right around 40 minutes, it looks like there's more p53 with the drug.
Right, I think that's right. So if we start looking here, they kind of look similar through 20, but at 40, there's pretty, just eyeballing it, a little quantitative down here, but just eyeballing it, there seems to be a lot less here than here, same thing at 60. There seems to be more still around when you keep the drug there than if you take the drug away. I think I would like to have seen this a little bit earlier too, but they then say, okay, let's not eyeball it, let's actually quantitate it, so there are ways to take these bands on a gel and quantitate it through densitometry. And so here's, I think, a much less subjective way of looking at it. Okay, so the open symbols are with the drug, with continued drug, and the black circles are without the drug, or when you took the drug away. So here I think it's pretty clear what we were eyeballing up here seems to be correct. So with the drug, there's more p53 around, so it looks like part of what this drug might be doing is extending the half-life of p53. Right? So instead of 20 minutes, maybe now it's 40 minutes, it survives for 40 minutes or something like that, on average. So that, I hope, fits into our bigger picture, right? p53 lasts longer before it's destroyed, therefore it has more opportunity to do what it's supposed to do, which ultimately, hopefully, is kill these cancer cells. Right? It's all starting to fill the picture. Good? Okay, so I think this is a very nice experiment. And then since, of course, we're looking at acetylation, and we know it affects the acetylation of histones, the next question that they ask is, does this affect the acetylation of p53 itself? So what they have here is kind of a similar, generally speaking, it's a similar kind of experiment that we've been looking at. They have antibodies against p53, and we're going to ask the question, is the acetylation levels different with and without drug? But apparently they're, and acetyl groups, which I guess we should have drawn earlier. All right, that's an acetyl group. Tend to, when they're added onto proteins, tend to get added onto lysines. Which in the single letter abbreviation system is K. So there are 3 distinct lysines in p53 that can pick up an acetyl group, or an acetyl group. And they are lysine 320, lysine 373, and lysine 382. And you can make a distinct antibody that will pick up only that one, only that one, only that one. So we can distinguish the 3 different, whether each of those places picks up an acetyl group on p53. Okay, so that's kind of the setup. Let's see what we got here. Okay, so this bottom one again is sort of just the control. It's just any old P53. Okay, so that looks pretty consistent. So again, that's our control. These other ones are antibodies specific for the acetyl group on each of these three locations, so 320, 373, 382. Okay, so they're doing this as a time course, so everybody gets, I'm sure, the 10 micromolar drugs. Right, so everybody got the 10 micromolar exposure to the drug over 0 to 24 hours. Again, here's our other control with the SAHA drug here for 16 hours. This is a comparator. So, what do we see? Let's go through each one. So, what happens to the acetylation levels, I guess, on lysine 320 over time? Increases starting when?
8
Yeah,
hours.
there's kind of not a whole lot, maybe a little tiny before, but definitely by 8. And it seems, again, just eyeballing it, kind of consistent after that. So by about 8 hours, definitely there's some acetylation on the lysine 320. How about the 373? It's exactly the same thing.
Starts with a
Increase of 1.
Yeah, definitely gets started earlier. There's a lot more here than here. But again, seems to start peaking at 8, but a lot more at, let's say, 16 and 24. And then how about the 382, third one?
Starts immediately.
Yeah, there's definitely a lot there, even under normal circumstances, more at 1 hour. I think this one's kind of a little weird. Looks like it goes down a little bit, then goes back up. So this one seems a little less straightforward. Okay, good. Okay, so that's that. So ultimately, okay, the three groups or the three acetyl locations seem to not be treated equally. Okay, they're not all the same. Okay. And what they're suggesting is that, you know, sort of their ultimate model is that adding and maintaining acetyl groups on one or more of these locations is what's extending the half-life and therefore extending sort of the potency, I guess, of P53, right? Again, if P53 is around longer, it can exert its ability to kill those cells more effectively. So one good way to try and show this is to start doing start creating mutant versions. So they construct mutants in each of those three locations. And I should have looked that up for R was. Shoot, I didn't. But you know off the top of their head what R is? What amino acid R is? That's one I don't know off the top of my head.
Does anybody have one?
Oh, I
What
just-
is book?
I just forgot. I meant to do it before I got here and I forgot. Maybe I can do it faster than me then. I want to say it's arginine, but I don't know that for sure. Is it?
I didn't say that.
Oh, there it is.
Yeah, I told you.
It is arginine?
Yep.
Okay, good. Okay, good. Okay, so they changed the lysines into arginines. Okay, so they have mutant versions of each of those. And then they did them, as you can see here, in all possible combinations. So they have versions with just one mutation. They have versions of the protein with 2 of the 3 mutations, and then they have a mutant version with all 3 changed into arginine. So they really were nice and thorough. It doesn't seem to affect the expression. Okay, so it's still there, and there's our beta-actin control. Okay, but what they are, what they want to look at here, they then used these mutants in terms of that luciferase experiment. I guess I should not erase that, okay, but you remember that, right? So they're looking for the mutant's ability to hop onto that promoter and turn on that luciferase gene. And here we go. Okay, so here's the wild type, so here's our control. Okay, so no treatment is the dark line. The other line is with exposure to that drug, the new drug. Okay, so here's our fully wild type P53, lots of luciferase, just as we would expect. Okay, so now we're going to compare everything else to that. So, here we go.
So we have the second most, one of these luciferase with a mutant in 320.
Okay, okay, so what would that suggest to you about that particular location vis-a-vis the ability of P53 to do its job?
It's a higher location for MCO group to be attached on, P53.
Say it again?
It's a higher area for the single group to be attached to P53.
You think? What does this mean? The mutant's still working well or not working well?
Well, that's the drug, right? So,
These are all in the present, everything's in the presence of drug.
right, the gray, like the light gray bar.
The gray box, yeah.
Okay, so.
Yeah, the no treatment we don't have to worry about.
So,
So does this mutant work well or not? Not as well, but
still pretty
pretty
good.
well. Okay. So what does that tell us about sort of the importance of that site being a lysine? Since it's not a lysine anymore, it's an arginine.
That's pretty good.
Not that important.
Not as important.
Right, it's not that important.
You're making it a-
Well, it's not a question of making it or not making it. We already know they're all being made. That's what this told us. The question is, maybe I shouldn't have erased the picture.
It's being expressed. That's
No, well, what being expressed?
good.
I need a,
The P53.
no. Again, part B shows us, we already know, all of these mutants get expressed.
Okay.
What we're asking is, right? Can the mutant still bind to that response element and turn on the luciferase gene?
And yes.
Seems to be able to.
Yeah.
Fractionally not as well, but relatively speaking, pretty well. So if the mutant does the same, close to the same thing as the wild type, what does that tell us about that mutation?
That's not really important.
Not really important. Okay, do we see that?
Yes.
Okay. So that might not be so important, at least singly, right? Because that's just the single mutation there. Okay. So we're going to keep doing this. Let's stick one right up. Okay. What about the second one? What about 373? Seems to be more important because this is maybe about half eyeballing it as that one. So that one seems more important. How about the 382?
Very important.
Seems pretty important. That's not very much compared to the wild type or the other two sites. So we seem to have this sort of in backwards order of importance, in a sense. And they started doing doubles and triples. Okay, so here's the first two sites mutated, but the third one's okay. Okay, that one kind of puts it back, it's a little bit higher. Okay, but all of the the combinations where the third one is mutated are not as good as the others. So I would say collectively this is showing that 382 site, that 382 lysine seems to be of the three the most important. And anywhere where that is a mutant site, okay, is our four lowest numbers here. So that seems to be an important site in terms of If you can't assimilate that particular lysine, then perhaps the P53 is not lasting as long and therefore not being, in this experimental situation, not being able to turn on the luciferase gene very well. That kind of makes sense? Okay. Or you're just nodding because there's only 5 minutes left of class. Okay.
There's one question.
Yep.
I was kind of confused about why the triply mutated one was more functional than the one with the single mutation you created.
Good question.
Is there another lysine perhaps, or that it binds?
I don't know the primary structure of P53, so I can't answer that question. I don't know the answer to that question. But it's a good observation. I don't know the answer. But it's a good question. Okay. And then the final experiment. They're looking at acetylation at that site, so they kind of conclude like we did, that the 382 site is the most important site. Again, in cell death, so they're looking, it's kind of the same experiment here, except the bars are now meaning surviving cells. So no treatment, right, everybody survives, not that surprising. Cells that are exposed to the drug, okay, versus the various sites of mutation. What do we see?
We see that with the last mutation, the cell survival is, I mean, with the K382R, it's the lowest survival within mutants.
Lowest survival. I would disagree with that.
No, they all survive.
And there isn't a mutation in 382. The
Right,
cells,
there's no mutation, that's what-
that cells, there's not a lot of survival. Or when 320 and 373 are mutated.
Right.
Cells
So,
survive.
yeah, that's how I was thinking about it. So when 382 is mutated, more cells are surviving, and these are cancer cells, this is a bad thing, right? Okay. So if you mutate that site, that particular lysine, you take that away, what can't happen at that site? Oh, yes, what chemically can't happen at that particular lysine is mutated. Right, it can't be acetylated, right, because it has to be a lysine in order to pick up that group. So if you change it to an arginine, it can't get that group, right? And therefore, if it can't get that group, it's either, well, it's probably a combination of not being able to work very well, and or it's not surviving. The P53 protein as a whole is not surviving as long because that seems to be an important place where an acetyl group is very, very helpful. Right? Perhaps in 2 ways. One, it makes it last longer. If it lasts longer, it can do its job. Or it's blocking the ability of it to do its job, which is jump on these genes that turn on apoptosis. I've just, I think, summarized the rest of the paper. Okay. So we saw in the presence of the drug, we kept acetyl groups on histones and P53. That in turn affects the levels of MDM2 and CYP21. We saw that the drug increases the luciferase reporter experiment because P53 is lasting longer, right? So that kind of links together. Okay? And that 382 lysine seems to be the most important one, perhaps again as a stability idea, and more directly, perhaps, it's changing the ability of it to actually bind to its target genes. So, given that, this could perhaps, under certain circumstances, be a good drug. 30 seconds to spare. This is not an easy paper. Part of the reason I chose it.
The last slide, figure 6.
sorry, not this line. So you're saying if you remove, 382, P53 can't bind, and that shortens the,
And if P53 can't bind to its
then it's not gonna,
genes, what doesn't happen?
it's not fixing,
We're not looking at cell survival, so.
cell survival decreases. Or increases, sorry, or increases.
And if you're a cancer cell, that's a good thing,
Okay.
right? So, mutating that site, that particular lysine, gets you to survive longer. That's pro-cancer.
Okay.
Right? And what they're trying to figure out is why. Why 381 and not 1742? Well, because that's a particular lysine that when you mutate it, can't get an acetyl group, and when you do the rest of the paper, that's changing half-life, that's changing stability, that's changing perhaps the ability, basic ability of P53 to act as a transcription factor. That makes sense. I think we need to spend more time on this paper. Let's see. Okay, now, our next test, which I don't think is pointed,